A series of technical reports and regulatory documents has reignited a heated question. Where do modified mRNA vaccine components travel inside the body? And were those data handled transparently? Hello, this is Dr. K. Paul Stoller, and I will break down the key claims and why they matter. Researchers and regulatory critics argue that early preclinical studies show lipid nanoparticles and modified mRNA don't stay at the injection site. Instead, the materials can appear in organs like the liver, spleen, adrenals, and, in some datasets, ovaries. The reports contend that some of these findings were redacted, edited, or downplayed in documents submitted to different regulators. Independent reviewers compared versions of biodistribution studies filed with Japan, the EMA, and later released by the FDA. They identify 4 troubling patterns: images that appear cropped or altered, graphs with critical data hidden, an initial high-dose animal experiment that showed toxicity but wasn't widely disclosed, and sex-specific signals, particularly spikes in female tissues, that were not clearly reported to all regulators. Authors describe these as potential manipulation or improper transparency. Normally, biodistribution studies, which show where and how long a biologic persists, inform safety reviews before human trials. The reports argue that for mRNA products, some studies use surrogates or non-standard methods, and that certain analyses were not done to internationally accepted standards prior to emergency authorizations. At recent advisory meetings, company and regulator representatives offered differing explanations. Critics say admissions at those meetings and later document releases suggest coordination between sponsors and regulators in how data were presented. Company spokespeople and agencies have disputed various characterizations. Meanwhile, independent analysts have published detailed line-by-line concerns. Why does this technical debate matter to the public? Biodistribution affects safety assessments, dosing decisions, and population recommendations, especially for groups like pregnant people and adolescents. Transparency in preclinical data underpins informed consent, trust, and the ability to detect rare or sex-specific risks. The reports call for full disclosure of raw preclinical data, independent reanalysis, sex-specific and long-term studies, and clearer regulatory standards for gene delivery technologies used as vaccines. They urge policymakers to ensure regulatory reviews are rigorous, independent, and publicly accessible. The debate is technical but consequential. As a society, we are still not at the point where we are able to recognize we were and still are the victims of a democide. So we are still having to ask regulators for the transparency and independent review that scientific trust requires. We should be at the point where we have Nuremberg-level trials. One day, perhaps, but not today. We will just have to keep asking nicely, even if that requires lawsuits, for transparency and for regulators to do their job and actually be regulators and not facilitators of the ongoing democide.